Hello, everyone, and welcome to Evidence Mastery. I'm Dr. Massoud Toussi. Today we're going to tackle a really critical question. How can you actually prove a drug works when a traditional clinical trial just isn't possible? We're going to do that by breaking down the FDA's latest guidance on this very subject.
So what we're looking at today is the FDA's draft guidance on externally controlled trials. Now, this is a pretty dense document, so we're going to be hitting the key highlights. I always recommend reading the full guidance to get the complete picture, but this explainer will give you a really strong framework for understanding it.
At its core, this whole guidance document tackles one fundamental challenge. How do we show a drug's effect when a traditional randomized control group isn't feasible, or maybe it's not even ethical? Well, that is the exact problem that externally controlled trials are designed to solve. Okay, so here's our game plan. We're going to cover 5 key areas. We'll start with the basics, what these controls even are. Then we'll get into the really crucial design considerations. After that, we'll talk about data sources, how to analyze everything correctly, and finally, how to navigate that all-important regulatory review. So let's dive right into Section 1 and get our definitions straight. What exactly is an externally controlled trial, or ECT, anyway? Well, to put it simply, it's a trial where you're comparing your patients on a new treatment to a control group that wasn't actually in the same study. Now, this control group could be historical, meaning you're pulling data from the past, or it could be concurrent, where the data is collected at the same time but from a different place, like a patient registry. And that brings us to Section 2. Now, this is arguably the most critical component of the whole thing, the study design. Getting this part right is absolutely fundamental to whether the entire study will be seen as credible. A major theme you'll see throughout the guidance is the need to minimize bias right from the start. And that means everything has to be pre-specified in the protocol before you even think about starting the trial. We're talking about which data sources you'll use, your eligibility criteria, your endpoints, and your entire statistical analysis plan. No wiggle room. This brings us to one of the biggest challenges, confounding. This is what happens when some other factor gets mixed in and distorts the results, kind of muddying the waters. For example, what if sicker patients are more likely to get the new drug? Their poor outcomes might have nothing to do with the drug itself, but just their underlying health. Without randomization, teasing these things apart is a huge hurdle. Now here's a really tricky design challenge that the guidance points out. How in the world do you define a consistent starting point, what we call time zero, for absolutely everyone in the study, both in your treatment group and your external control group? This starting point is officially called the index date. And if it's defined differently for the two groups, you can introduce a massive bias right from the get-go, which can make your whole comparison totally unreliable. This can lead to a very specific and pretty dangerous trap called immortal time bias. Just imagine your study requires patients in the treatment group to survive for 30 days just to be eligible for the drug. Well, by definition, you've selected for survivors. If your control arm doesn't have that same 30-day survival requirement, you're not comparing apples to apples, and it can create this false appearance that the drug is way more effective than it really is. We also have to think about assessing the outcomes. You see, unlike in a blinded trial, here, everybody knows who got the treatment and who didn't. That knowledge can subconsciously influence how outcomes get reported or measured. To fight against this, the FDA really stresses that you need to use objective endpoints, things like lab values or survival, that are measured in the exact same way across both groups. So that naturally leads us to Section 3. Where does the data for this external control group actually come from? And maybe more importantly, how do we make sure it's even comparable? Broadly speaking, there are 2 main buckets for this data. On one hand, you could use data from previous clinical trials. That data is usually really well structured and clean, but the downside is it might be a bit outdated. On the other hand, you can use real-world data from sources like electronic health records. This data is very current, but it's often messy, inconsistent, and can have a lot of missing information. The guidance gives us this fantastic checklist for making sure our groups are comparable. You have to ask these really critical questions. Has the standard of care for the disease changed over time? Are the patients in both groups at a similar stage of their disease? Are they getting similar background treatments? And so importantly, are the follow-up times and the outcome definitions absolutely identical? Any little difference here can completely sink the comparison. Ultimately, the success of the entire trial really boils down to this one principle. The external control group has to be as similar as humanly possible to the treatment group. Every single difference between them introduces another potential source of bias that can undermine your results. So you've got your data. This brings us right to Section 4, analysis. Once you've designed the study and you've collected the data, how do you analyze it while accounting for all that inherent potential for bias? And guess what? You're going to hear that key phrase again, pre-specification. The statistical analysis plan, or SAP, has to be completely locked down before you look at any outcomes. This plan needs to detail everything: your endpoints, your methods for controlling confounding, how you're going to handle missing data, and even the sensitivity analyses you'll use to stress test your own conclusions. The guidance also gives some really practical advice on when you should even think about using an ECT. They're most convincing when you're expecting a large, really dramatic treatment effect, like a breakthrough cancer drug. Trying to detect a subtle, small effect with this kind of study design is incredibly difficult because it can so easily get washed out by all the statistical noise and bias. And that leads us to our final section, the practical side of things. What are the steps for engaging with the FDA when you're proposing one of these externally controlled trials? The FDA makes their expectations crystal clear. First, talk to them early in the development process. Don't just spring this design on them late in the game. Second, you have to be ready to robustly justify why a traditional randomized trial isn't feasible or appropriate. And third, and this one's a big one, you have to make sure the agency has full access to the patient-level data from both arms. For them to validate your findings, total transparency is absolutely non-negotiable. This quote from the guidance really just sums it all up, doesn't it? The purpose of conducting clinical investigations of a drug product is to distinguish the effect of a drug from other influences. That's the challenge of all clinical research, and as we've seen today, it's particularly difficult, but definitely not impossible, to overcome in an externally controlled trial. And that is our whirlwind tour of this very important FDA guidance. These concepts are all part of the much larger landscape of real-world evidence. For anyone who's interested in exploring these topics even further, I've covered them extensively in my upcoming book, Real-World Evidence, Principles and Practice, which is going to be available in May 2026. I'm Dr. Massoud Toussi. Thanks so much for joining me on Evidence Mastery, and I look forward to seeing you next time.